Welcome to part 2 of this short series on different CHIPS2 tools for microbiome data tidying, visualization, and statistical analysis. In this video, we will focus on tools for inspecting and tidying our data so that we can ensure they are fit for downstream analyses as part of an analytical workflow. The first tool to consider is one called Sequence Numbers, Rarefaction Curve, and Alpha Diversity Estimates. I have two images on this slide. Let's start with the one on the right. This image is a screenshot of a text file produced by the tool. On the top, we have a list of sequence IDs like F3D0 and F3D1 and associated numbers of sequences. Depending on the community profile, the numbers of sequences range from 2,629 to 16,662. This type of variation in sequencing depth is typical to microbial community datasets. And on the bottom of the screenshot on the right, there is a list of alpha diversity estimates. Alpha diversity is a term used to refer to within community diversity. The first column contains unique IDs, and only a smaller section is shown to better display the data on this slide. Moving from left to right, we have several other columns, starting with observed numbers of OTUs. We also have columns for the Chow-1 index, which is an estimate of taxon richness based on abundance data, the Shannon Diversity Index, which takes into account both taxon richness and evenness, and Pillou's evenness. Here, richness pertains to the number of different taxa in a particular community, while evenness pertains to the relative abundance of different taxa within a community. Low evenness means that certain taxa in the community are more dominant than others, while a higher value would correspond to a more even distribution of taxon abundances. Diversity is influenced by both taxon richness and evenness. When richness and evenness increase, generally so does diversity. The tool then also provides an option to tabulate some selected phenodata variable together with alpha diversity estimates to make the data easier to read. In this case, the data have been tabulated together with the group column in the phenodata table. Then we have the image on the left, which is a rarefaction curve. This is a line plot with numbers of sequences on the x-axis, with increments of 5,000 sequences. The values range from 0 to over 15,000. The y-axis has numbers of observed OTUs with increments of 100. The values range from 0 to 400, and several lines are shown on the plot corresponding to different microbial community profiles. These types of plots are used to visualize the relationship between sampling effort, in our case the sequencing depth, and the number of OTUs. If the lines approach the asymptote, this means that there are no more new taxa to discover. In practice with microbial datasets, that asymptote is very rarely reached. These types of plots are also useful for visualizing differences in sequencing depth within a given dataset. So in our case, certain communities have close to or over 15,000 sequences, whereas others have far less. So variation in sequencing depth is a source of uncertainty in microbial community analyses, and there are many different ways to deal with it. One option that has been traditionally used involves rarefying to even depth, that is, subsampling the data to achieve a common sequencing depth for all community profiles. As you can imagine, this can result in the loss of considerable amounts of data. Rarefying to even depth is possible in CHIPS2, but it should be noted that there's also considerable evidence for drawbacks to this method. If you're interested in finding out more, I would suggest looking up a very often cited paper published in the journal PLOS, Computational Biology, in 2014 by Paul McMurdy and Susan Holmes. And the paper is called Waste Not, Want Not, Why Verifying Microbiome Data is Inadmissible. Chipster also offers alternatives for verifying to even depth. These include particular ways to transform data and internal corrections for unequal library sizes used by the DSeq2 package. The next thing to consider is the available selection of taxon level cleanup tools. First of all, it is possible to produce an overview of the taxon composition of a particular phyloseq object at some user-specified level. So for example, the phylum level. There are also tools for removing undesired sequences from the dataset, for example, by retaining only those sequences that have been classified as bacteria or archaea. It might be surprising, but many microbial community datasets also include chloroplast and mitochondrial sequences, and it is possible to remove these if they are not of interest. If, when using the taxon composition overview tool, you discover some particular taxon that you'd like to remove, that is possible by manually entering the name of the taxon you want to exclude. And overall, these functionalities in Chipster are divided into a set of 3 different tools. So options for tidying microbial community dataset sets in Chipster further involve some tools for visualizing and filtering out low abundance OTUs. There are 2 approaches to this, a prevalence-based one and another based on the abundance of singletons and doubletons. Prevalence refers to the number of samples in which a taxon appears at least once. In Chipster, it is possible to visualize taxon prevalences and apply a prevalence filter using a user-specified % threshold. There are also options for tabulating singleton and doubleton abundances as a text summary, and for removing singletons and doubletons from the phyloseq object. The figure shown on this slide is a prevalence plot that can be used to visually estimate a suitable prevalence filtering threshold. The x-axis shows total read numbers displayed logarithmically with values of 1, 10, 100, and up to 10,000. The y-axis corresponds to prevalences shown as fractions in relation to total sample numbers. The y-axis values range from 0 to 1 at increments of 0.25, with 1 corresponding to 100%. So, for example, if a given taxon has a value of 0.05 on the plot, it occurs in 5% of all samples included in the dataset. Within the plot, there are several panels based on taxonomic classifications at the phylum level. In this case, proceeding from the top left toward the right and then downward, we have panels for actinobacteria, OTUs that are unclassified at the phylum level, Bacteroidetes, Deinococcus thermis, Firmicutes, Peteschibacteria, Proteobacteria, Tenericutes, and finally Verrucomicrobia. Each dot on the plot corresponds to a separate OTU. There's also a dashed line toward the bottom of each panel. This corresponds to a prevalence filtering threshold of 5% that is included in the prevalence plot as a guess. So in practice, when performing prevalence filtering in Chipster, the user may also set other values as the threshold. So that's all for this video, and the next video will contain information on transformations and ordinations. So I will see you there.